get started and welcome everybody to the last Precision Medicine Symposium for the year. And I think we're going to reconvene in September, and we would love people's opinions about whether we go back to completely in person, whether we do hybrid, or how we do it. We argue back and forth, and there's advantages and disadvantages. But from the standpoint of our speakers, many of whom come from a long way, it gets kind of lonely up here with less than 20 people. So today, we've been able to entice Sam Sternberg to talk to us. I will let him introduce himself, which is what he prefers to do. I do want to mention a few things that actually Sam went to Columbia and graduated in 2007, then went and did his PhD at UC Berkeley. And I have to say this, the first time I actually knew of Sam's name was the book that he and Jennifer Doudna wrote, and I wrote the top, it's called A Crack in Creation, Gene Editing and the Unthinkable Power to Control Evolution. So from that standpoint, he's famous. So Sam, thank you very much for joining us to do it.
I appreciate it. All right. Thanks, Ron. Thanks, Sheila. I'm gonna see if I can hide this little display at the top.
People on Zoom are not seeing this, it's just us.
Just, they're not seeing the slides?
They're seeing the slides, but not this two versions.
Oh, but for us, I'll get rid of it too. I just go floating in controls. All right, and admits. Okay. I told Sheila or Driti that I wasn't exactly sure who to make for this talk was going to be. So, as I always say, I'd love to be interrupted if I'm not hitting the right level of detail, or if you have a question. It's an informal group, so I'd love to keep it informal. And let me see if I can get my laser pointer. Great. Okay, I'm going to talk about a new type of CRISPR today, not CRISPR. Oops. Sorry, let me try to fix this. Okay, it's gone. Not CRISPR nucleases, but CRISPR-associated transposases that we've been studying and developing in my lab for a new type of gene editing approach involving DNA insertion.
If you go to the more and tell it,
Yeah, I just did that,
just do it again. Do it again. Go to the more. Because every time somebody comes in, it does this.
Can we? Is there a way to like not
Just
have,
do hide.
Yeah. I know how to do that,
Good
but
luck.
I don't know why it comes back up.
Good luck.
Okay. Introduction. So I came from a CRISPR background that was motivated by this biological question, which is how bacteria defend themselves against viral infections. When I joined Jennifer's lab, this was back just a year or two after this foundational study first discovered the role of CRISPR in providing an adaptive immune pathway that is ubiquitous in both bacteria and archaea. And so when I started my lab here, we were really broadly interested in further probing the diversity of these immune systems, thinking that they would continue to provide opportunities for building new genome engineering tools. This is a slightly out-of-date tree of life, if you will, for CRISPR-Cas systems. These are actually far more diverse than the Cas9 enzymes that you're probably all familiar with. So Cas9 that is used widely in gene editing is this 2A system in the top right. There are dozens of other subtypes, most of which rely on other enzymes that are not Cas9. The kind of unifying hallmark feature that's true for all CRISPR systems is reliance on a guide RNA that provides the information on where in the genome to target, but the downstream execution upon targeting can be very different. Can be DNA cleavage, it can be a single double-strand break, it can be a DNA nick. And I'm going to talk about a new kind of chemistry today, which is a DNA insertion. But broadly, we think that by continuing to look at this biodiversity, we'll probably find things that still have yet to be discovered. So conventionally, we think about CRISPR as existing in bacteria to recognize and thwart invasion of foreign nucleic acids by viruses and plasmids. And like I just said, there are diverse nucleases that are used by different kinds of systems for this purpose. Here's my little animation for non-scientists, or maybe folks that are more on the clinical side, just to highlight what is actually going on in bacteria during a CRISPR immune response. And again, it was the study of this process that really revealed the molecular mechanism of Cas9, which was the starting point for the development of this transformative gene editing approach. And so after a phage or virus infects its DNA, CRISPR complexes form between the enzyme and a single or two-piece guide RNA, whose job is basically to surveil the cell for those invading molecules of DNA, recognize those DNA molecules through base pairing, and then target both strands of the DNA for DNA cleavage. And so obviously, I don't need to stress here that by studying the molecular details of this adaptive immune response, there's been an explosion in this CRISPR toolkit, which is far more than just the molecular scissors. That's where we're going to talk about kind of the newer tools today. And we're thinking about continuing to use those as now technologies to engineer the genome. And I'll just point out here that I think it's not a surprise that with restriction enzymes, with CRISPR enzymes, we continue to find huge opportunities in looking at this viral immune response, because the same properties that immune systems need to encode, namely a high degree of specificity for their targets and distinct mechanisms of action, those are the same kinds of molecular properties that are very, very useful for building tools. Okay, so on the left, we have the outcome of a DNA double-strand break. It generates a point or a trigger point for further degradation of DNA. That's why DNA cutting in a bacterium during a viral infection leads to protection from that viral genome. Obviously, if you now carry out the same reaction in eukaryotic cells like human cells, we're now tapping into this ability, thanks Rodney, for double-strand breaks to be a trigger point for various forms of DNA repair or recombination. And so in this very simplistic view, the double-strand break is harnessed together with the repair template for now instead installing a permanent genetic modification at the target site. And where CRISPR really transformed gene editing was not the knowledge that double-strand breaks could be used for this purpose, but the ability to easily reprogram these molecular scissors to target anywhere in the genome simply by changing the sequence of the guide RNA. And so I'm not going to really talk about patient-specific applications today. We're going to go deeper into the CRISPR biochemistry. But again, not knowing who my audience was today, I wanted to just make sure we're aware of some of the amazing opportunities to harness these systems as a clinical approach for both genetic disease and cancer. And so broadly speaking, many of the ongoing clinical trials now are either editing patient cells ex vivo using various ways of delivering CRISPR enzymes into those cells before an autologous transplant back into the patient. And then I think some of the more challenging targets are going to involve in vivo delivery to actually correct mutations directly in the body. And so this was a study that's now almost 7 or 8 years old. Actually, in the pre-CRISPR days, this used an older gene editing technology, but was used on T cells from this 1-year-old girl, actually T cells from a donor in a form of immunotherapy that ended up curing her, or at least getting her healthy enough to receive a bone marrow transplant. And then Victoria Gray is the first US patient that's been treated with gene editing drug for sickle cell disease. And in the latest clinical results from Vertex, they've now seen, I think, a 90+% transfusion-free rescue for all the patients that have been treated. So we're really at the cusp, I think, of, for certain genetic diseases, a new era of genetic medicine. The question is, can we continue to build out the CRISPR toolkit to solve some of the more challenging targets? And so I've shown this slide already, and I'm going to pivot to some of the work that we've been specifically focused on in the lab, which goes kind of deeper into the CRISPR biology and focuses on new connections between CRISPR systems and transposons, which I'm gonna hope to convince you are revealing new opportunities to harness CRISPR-associated transposases for gene insertion.
Sam, you might want to just X out that
Yeah.
stupid thing.
Thank you, Rodney. Is it the same on both sides? Where's
Yeah.
my mouse?
It'll be on your screen too, yes, that is. Okay. Don't worry about that.
Okay. All right. Okay, why transposons? So my lab, we started really as like a CRISPR lab, and I think we're increasingly getting obsessed with transposons. Just to get us all on the same page, transposons, of course, are selfish genetic elements, often called jumping genes. Oh my goodness.
Just those go away fast, don't they?
Okay. Transposons are these minimal pieces of DNA that, in their simplest form, encode a gene called a transposase that is an enzyme with the capability to recognize and mobilize a piece of DNA bounded by inverted repeat ends from site A to site B. Mechanism for a cut and paste transposon. Again, here's the mobile piece of DNA. The transposase gene is expressed into these enzymes, which recognize the inverted repeat ends. The enzyme is represented by these spheres. The ends get synapsed together, triggering excision of the transposon DNA from its donor site, and then reinsertion into a new target site, which could be in the same genome. It could be a vector, it could be a plasmid. We could be integrating DNA from a plasmid to the genome. That's quite up to the experiment. But point is that this piece of DNA encodes all the necessary machinery to perform this DNA integration reaction. We're quite fascinated by transposons and transposases for a number of reasons. One is their link to CRISPR, which I'll get into, which maybe isn't surprising if you consider that this gene family is by far the most abundant gene in all of nature. This is like, I would say, nature's most successful primitive gene because it contains the instructions to make more copies of itself by spreading it around genomes. And it does so in very diverse mechanisms in collaboration with diverse kinds of different components, one of which is CRISPR. In a human capacity, of course, transposons or retrotransposons make up a shockingly large percentage of the genome. I think it's actually now over 50% with the latest telomere-telomere genome assembly. And transposons have also been used widely as tools because they have some of these very useful properties. Number one, they've evolved to do this reaction on very large genetic payloads, gene-sized payloads. So I'm going to come back to some of the challenges with gene editing version 1.0, but transposases have evolved to move large genetic payloads. That's going to be a distinguishing feature. They're generally pretty autonomous in that they do this reaction on their own. They don't have a lot of requirements for host factors. And I'm going to come back to the importance of this third bullet point, but they generally do this reaction without leaving behind DNA double-strand breaks, which has implications for the response of the cell to the integration event itself. And I'm going to come back to that later. What has kind of limited the use of transposons is that if you look at the behavior of most transposases in terms of their specificity, by and large, DNA targeting and DNA integration is non-specific for most transposons that you'll find in nature. So let me give you an example of that. Here we took a transposon. This is actually from an insect. It's called Mariner. We expressed the transposase with the donor DNA in bacteria. We tested the DNA insertion specificity across a large population of cells by high throughput sequencing. And then we mapped on this plot across the E. coli genome, the site where any transposon was inserted across the population. And you can immediately appreciate that there's just a wall of Illumina reads that are tiled across the entire chromosome. There's really no pattern to this activity. It's just indiscriminately targeting DNA regardless of underlying sequence. And many transposases that are used in eukaryotic cells, like piggyback, like Sleeping Beauty, if you did this on retrotransposons, you'd see by and large the same type of behavior, a general lack of any targeting specificity. Now, against that backdrop, we got very excited when I saw a paper get published in 2017. It was a bioinformatics paper just looking at genomes. And there was a lab that found genomes that encoded what looked like transposons, where actually next to the transposase genes were CRISPR-associated genes or Cas genes. And in addition, there were CRISPR arrays. These are the schematic used to denote regions that produce guide RNAs in the cell. But there was one major surprising feature, which is that these CRISPR-Cas systems inside transposons have always were missing the nuclease component. So I told you all CRISPR systems use guide RNA, and they all work to protect the cell from infection by cutting. But these guys have lost the nuclease. Either the genes missing or the actual active site of the nuclease domain had conserved point mutations that made it very clear this enzyme will not be able to cut DNA. And so the question from the paper is like, what are these CRISPR systems doing that appear to have evolved without the ability to cut DNA, and why are they inside of transposons? And looking at these, it kind of became clear that, well, what you're left with is a module that has guide RNAs and proteins that bind guide RNAs that could still target DNA using RNA guides. And they're flanked by genes that encode enzymes to do all of that DNA excision and integration chemistry. And perhaps by working together, these CRISPR-associated transposons, or CASTs is the acronym I'll use for the rest of the talk, might have the ability to actually do DNA integration at sites in the genome that are specified by RNA-guided targeting. So the outcome here is not DNA cleavage to protect the cell. We presume that actually the transposon stole the CRISPR system and repurposed it as a way to specify where in the genome it gets spliced. So this was really interesting for kind of biological reasons. It was also exciting from technology reasons because, well, it would offer a potential solution to this accuracy problem. Now we've got transposases that might be targetable. If they're transposons, they probably evolved the ability to move around large genetic payloads. So now we have a way to move around kilobase chunks of DNA. And because they go through this transposition reaction, they're not going to make those DNA lesions in the process. So we were interested to kind of test this hypothesis because it was biologically interesting and had some clear opportunities as a technology, depending on if they worked the way we thought and how well.
Sam, before you go on. So the square triangles that you show in this picture, which is the target for this transposase, I mean the CRISPR
Yes.
part. So what sequence is that going to?
Yeah, great question. So in nature, what are the guides and what do they target? There are generally two classes of guides. There's always one guide that targets a conserved region of the bacterial chromosome. So it's clear that these have a safe harbor locus they target with the guide RNA.
Isn't that locus where they are?
Yes.
Okay.
Now, there's a really interesting reason why that was not detected, neither by me nor by the group that first found these in genomes through bioinformatics. And that's because there's mismatches between the guide and the target, and I could talk to you about that later. But yeah, one guide is thought to target a safe harbor locus. So these transposons, the cool thing about transposons, they're selfish in that like they don't directly evolve to benefit the host. They kind of have their own reason for existing, which is just whatever makes them spread better. But they also need to tune their cost on the host because by inserting, they're kind of like probably expanding the genome size. That's not necessarily a good thing. So they often package genes that have a fitness benefit for the host. And they target regions of the genome where they can lie dormant and not interrupt genes. So they use a guide RNA to splice into regions that are intergenic but highly conserved. And then they have other guide RNAs that target mobile plasmids. And that gives them, we think, although we haven't tested this in native settings, Bacteria often are exposed to mobile pieces of DNA plasmids that are spreading in native environments. So these often target plasmids that probably give the mobile DNA, the transposon, a way to get onto a conjugated vector that can get out of the cell and go transform other cells through conjugation. So that's the selfish part, just a way to get between more genomes to spread. But then once you're in a host that has the safe harbor locus, you kind of integrate silently in a region that won't impose any fitness cost, and you lie dormant there until another plasmid comes along that you have a guide RNA against, and you hop in, and now you've got a way to get back out.
Does it split the sequence that it goes into when it transposes?
I'm going to talk about that, yeah. Okay, so those are the reasons we wanted to study this. So this was one of the first projects in the lab led by integrated program student, Sana Klumpe, who just graduated last December. So again, I'm going to use CAST to refer to these CRISPR-associated transposases. This is actually the native genetic locus of the CAST system we first decided to go after. So we could easily identify those inverted repeat ends. These are basically telling the transposase what the bounds of the piece of DNA are that you're going to mobilize. All these genes in gray, not going to talk about, they're really interesting. They encode things like antibiotic resistance or viral defense. Those are the reasons why these probably exist, because the cells that inherit and get the transposon are endowed with protection against antimicrobials or viruses. So that's why the transposon exists, because it's providing that benefit in the cargo genes. But what we cared about were the CRISPR genes, the CRISPR RNAs, and the transposase genes. And so Sana basically identified the parts we thought we would need. We developed a cellular assay for testing, do these transposases integrate the DNA at target sites that match the guide RNA? So on the bottom, we have a vector that makes guide RNAs and CRISPR proteins. We have a second vector that makes transposase proteins, and then a third vector we call P donor that has a mini form of the transposon. And our goal is, can we splice that transposon from the plasmid into the genome? And so we designed guides colored in magenta to match complementary sites in the genome. We hypothesized that the DNA from the P donor would be inserted somewhere nearby. We didn't know where yet, but we thought if it splices anywhere nearby, we can use primers, pairs of primers that bind to the genome and to the transposon and amplify successful integration products by looking for the new presence of an amplification band. And so we were very excited to find exactly that result. This is like the publication version of the gel, but I remember distinctly getting this Slack message from Sana when she got like the first band on the gel, very exciting. And she had immediately done all the right controls, namely here we had the guide RNA that was matching the target site labeled here as a 1. If she kept everything else identical, but now scrambled the guide RNA, so non-targeting or NT control, we lost a band. If we omit any of these vectors, we lose the band. And in the control I'm not showing, if we make a single base pair change in the transposase gene to knock out the active site, we also lost the band. So this really required all the active machinery and perfect complementarity between the guide and the target site and the genome. Now, to Rodney's question, where exactly did the DNA get integrated? Naively, we kind of thought, oh, if it's like a Cas9 editing reaction, you bind the target and you splice inside of the target site. And so 1 here represents the target. It's a 32 base pair target that is base pairing with the guide RNA. And rather than seeing that get interrupted, the transposons actually integrated exactly 49 base pairs downstream of the target site. Now, coming back to your question and the safe harbor locus, here's where it gets very cool. The safe harbor locus they target is in a coding region. So it's in a highly conserved gene that is fairly invariant, but it doesn't interrupt the gene because of this distance. So imagine now that the gene overlaps with this target, the stop codon somewhere around here. Because of this distance rule, it inserts downstream. So even though it's targeting a highly conserved region, it's not interrupting the gene through the integration reaction itself. That's probably why it evolved this kind of distance separation between where the guide RNA matches and where the DNA gets spliced.
We can talk later, but recombination between one and the real guy, which will have that sequence in it, could occur. Maybe it does.
Inside the transposon.
Yeah, but we can talk.
And that's where, yeah, we should talk later. We did a lot of other experiments I'm skipping over, but one of the immediate things we wanted to do, and this was heavily influenced by developments in the genome editing field. I'm sure you've all heard about, studied, suffered from off targets. So it's easy to look at one place with PCR and see a band that looks clean, but we wanted to make sure, like, it could be that these are still integrating randomly all over the place. And so we developed this high throughput sequencing approach that would be agnostic to location. We just capture any insertion event, regardless of where it occurred. We deep sequence those insertion events, and then we can extract the fingerprint that tells us the genomic site where it inserted and map those back to our reference. And I've actually already shown you some of this data for Mariner, which I told you integrates indiscriminately across the entire length of the chromosome. Here's now the same experiment under the same expression conditions with this CRISPR-associated transposase. The guide RNA matched this site shown here with the red triangle, and you can see now over 99% of the reads across a large population of cells, all mapped directly to the target site. And so this told us that these are fundamentally different than most families of transposase, which are non-specific. These guys seem to be completely dependent on the guide RNA for defining the integration site. So cartoon of how we think these work. So the first step in this pathway is very similar to the way that a Cas9 defines target sites for DNA cleavage and editing, which is base pairing between the guide RNA or CRISPR RNA and the DNA. This depends on a PAM sequence being nearby. But again, the CRISPR component here, which is called cascade, has no nuclease activity, which is why we're not getting any DNA cutting. Instead, we actually discovered that it brings along a new component called TNIQ. And the importance of TNIQ is to create a protein interface that directly recruits the transposase enzymes, which drag along the donor DNA. And we think it's actually the molecular footprint of each of these components that explain exactly why there's that strict disk spacing. There's basically 49 base pairs protected by these proteins before the transposase has access on where in the genome to splice the DNA.
So are you going to tell us about the 1% that are off target and how close they are to the off target sites? And
yeah, that's so funny because I literally met with someone this morning and acknowledged that I've been asked this question over the years and we still haven't really mined our data for that.
Okay.
So the answer is, like, in some cases, we can identify clearly similar off-target sites that are similar to the sequence of the guide. There could also be rare integration events that are CRISPR independent. And we have a project on a related CAST system, which is very prone to doing that. I'm going to come back to that actually in a couple of slides. But truthfully, we haven't gone deep into mining that.
Our loop areas. And actually there's some precedent in this field for wondering if just DNA deformation could be a hot spot for this reaction. So yeah, we've started thinking a lot about the kind of those types of mechanistic and sequence details, as well as also the structures of the different components. And I should have started by saying, you know, I'm a biochemist by heart, so I think a lot about molecular mechanism. One of the cool things is we teamed up early on with the cryo-electron microscopist Israel to take samples that Tyler in the lab had purified, where we had discovered using biochemistry this new co-complex between the CRISPR module and this new factor TNIQ. We guessed that it would be bound somewhere near the 3' end of the guide,
because of this distance rule. And then when we determine the structure, that's exactly where we saw TNIQ. I know it's a little bit jumpy on the screen, but everything in blue is the CRISPR complex. You can see the guide RNA is beautifully bound by all these Cas components, which basically splay out the base pairs or the bases of the RNA to promote base pairing with the target DNA. And then Q is stapled on the 3' end, exactly where it needs to be to recruit those downstream components. So on kind of the enzyme world side, the reason, again, we were really excited about this is that we think Cas are one of the first enzymes within a recombinase superfamily that catalyze truly programmable DNA insertion. And now I'm nervous saying that in front of Rodney because maybe I overlooked a category. But again, there are lots of transposases like piggyback, like TN5, or retroviral integrases that are, and now I'm scared of Steve Goff over here that now I'm making a mistake on that, but generally fairly non-specific. There's of course chromatin factors that are going to impact where these payloads go, but this transposase or integrase doesn't impart a high degree of sequence specificity. Then there are other integrative elements, things like viral integrases and phages, certain transposons like TN7, certain retrotransposons that are highly sequence specific, but they're not flexible. They have one place they always go. You can't like change it. And now we have a type of transposase whose very way of targeting DNA is programmable because it uses a guide RNA. And so we thought this could represent a really exciting platform for lots of different applications where the goal is putting DNA payloads that are large into genome at sites that we, the experimentalists, want to be able to easily control. Okay, so here's like a brief survey of some stuff we've learned over the years studying Cas systems in more detail. So one is that exactly like we would have expected, being programmable, we can easily change the site of the genome where we integrate the payload. This is just some representative data. Each graph here came from a different experiment in bacteria. The only difference was the guide RNA sequence. In each graph, I'm showing the site we hoped to target with the triangle, and you can see the blue peak perfectly moves exactly to where we target. So really easy to program these. Each of them are like 99%-ish. We should go and mine the other 1%, but we haven't really done that, embarrassingly.
So the Vibrio sequence is just super homologous to the sequence in coli, right? I'm talking about the original sequence, because you originally started this, you didn't know anything, but you said, gee, will this work in E. coli? Or did you change the sequence?
The guide RNA we changed to target LAC-Z, and all these are like arbitrarily chosen guides.
So the Vibrio guide, does it have a sequence in coli or not?
You know, it's possible that I've never looked at that. Cholera and E. coli, I mean, yeah, because it's a highly conserved, it does have the gene. I haven't looked if they're identical or not, but we never tested the native guide. We always just like clone a guide artificially to match whatever sequence we want to target. Yeah.
How is it induced? Is it continually expressed until it finds the target?
Great question. So in these experiments, we have inducible promoters that we designed to control expression. And we typically like put these on a plate, we grow it on solid media, and we induce them overnight. In nature, there's a big black box on what's the native regulation of the transposase in like Vibrio cholera, where it's naturally encoded. And like there's been a major gap in our knowledge about the biology of these transposons in nature. Like a lot of it is abstracted from the biochemical properties in our artificial E. coli overexpression system. At some point we brainstormed, like I'd love to go and find an ecological environment where these exist naturally, and then go do longitudinal sequencing. And can we actually watch it spreading between cells? And can we look at the native expression patterns and see if it gets induced? Like maybe a plasmid comes in, there's some stress response, the gene expression goes up, and now they're like primed to go hop into that. But that right now is total black box. And for the most part, everyone that's now working on this is doing the same type of approach as us, which is like put it on synthetic promoters in plasmids and just jack it up to the max to like study how it works or how to use it. Actually, that's another thing that literally was discussed for 30 minutes this morning in a meeting. And what came up in that meeting is it general toxicity from protein overexpression, or could there be specific toxicity associated with these enzymes? My guess is it's just general overexpression toxicity, because we do a lot of recombinant protein expression in human cells and in bacteria, and like I don't think there's a rhyme or reason when we see evidence of toxicity. But certainly for our use of these as tools, we're thinking a lot about, are we putting the cell in a state of stress by virtue of generating a large amount of enzymes that can integrate DNA? So I'd say that's kind of like sometimes, but we don't know if it's related to the transposase pathway or just general. This slide is just showing that we started with one candidate system in the CRISPR field, especially for porting systems from bacteria to other cell types. It's often the case that you need to start that kind of an effort with lots of different shots on goal. So we are able to use further bioinformatic mining, generate large phylogenetic trees of different caste systems. And then in this study, we just showed that we could pick and choose a few dozen. Some of them are highly active without any optimization. This graph is showing that we actually have a number of systems that are totally orthogonal, meaning system 1 will integrate the DNA associated with system 1, but not systems 2 through 5. So if we wanted to do fun experiments like mixing and matching programmable transposases to move around different kinds of payloads, we now have a cassette or a suite of different types of Cas systems that we can use. Rodney, this gets to your question about specificity. There are different families of Cas systems. Again, all of this is coming from the last couple of years of research. I'm not going to get into the details on the types, except to say there are two major types, type 1 and type 5. And they're actually pretty different in actually in two ways, but the way I'll talk about here is fidelity. Type 1, even if you zoom in on the lowest coverage on the graph, you'll still see very high accuracy for the target site. Type 5s, if you zoom in, you start to see walls of insertion events that look a lot like Mariner. And so these are not RNA guided. We have looked at these, and there's no rhyme or reason to where they go. The sequences are as random as if you randomly sample the genome and look for a match to the guide RNA. So these Cas systems, we think of two pathways. It's the story we're wrapping up right now. One pathway is RNA guided and RNA dependent. Pathway 2 is RNA independent, CRISPR independent. This just goes randomly like Mariner. And this could be relevant for the biology and nature, because one question that we've kind of thought about is it good to be highly accurate for a transposon to survive? You could argue that it's better to be random, because now you increase the chance of moving yourself around. And so that's something that we're kind of studying further with these systems.
What does SH stand for?
Cytonema hoffmanni, it's a
Oh.
cyanobacteria. We'll skip that. We've done more structures. I'll just point out here, since I got a nice smile from Rodney earlier, we've solved additional structures, and now we have even more precision on why that spacing is 50. So here's the CRISPR complex, here's that TNIQ protein, and now here's the latest component that we've determined structures of, which forms a ring. Beautiful. This is actually cryo-EM micrographs at the top. It forms a 7-membered homomeric ring that has a very prominent central pore. Beautiful, perfect dimensions for double-stranded DNA. We also have structures down the DNA. So this basically encircles the DNA coming out of the CRISPR complex. And if you put some measurements on how far you get from here to there, 50 base pairs. So basically, this is telling us this is how the DNA gets exposed and presented to the transposase for integration.
So what's the clamp loader for that?
That's a great question because this is in the same family of other AAA plus ATPases. We do not know jack really about how it assembles and disassembles. What's interesting is that this protein, ATP, is sufficient for it to form closed rings. So how does it open to get on DNA and how does it open to get off DNA? Don't know. It's undoubtedly going to involve ATP hydrolysis, but what the mechanism is, like, we don't know.
Sam, if you don't have PMIQ, do you have no activity or do you have random activity?
No activity for type 1.
So they need.
They
that
need,
trigger to go.
they need, yes, they need targeting by the CRISPR component. Again, that's not true for all systems. The type 5s, if you express the CRISPR complex without Q, I'm sorry, if you express the transposase without the CRISPR complex, or you just don't make Q, it'll go randomly. And it'll go randomly also with the CRISPR complex. It just like now has two different ways of moving, which is why you get on target and that spectrum of off targets. These guys that we've been studying, and there's a reason we've been studying these because they seem to have more optimal specificity because they seem dependent on CRISPR for getting to the target.
So I ask is the analogy that HIV integrase has lead-jet, which is kind of a crappy targeting, but it's also almost required for activity. So it's sort of doing both things less well than these guys.
Interesting. We've done experiments to show empirically that if we use these as a tool, and this is all here done in bacteria, they do in fact integrate payloads that can be tens of kb in size without any drop in activity. So here we just took our overexpression strain and we gave it transposon payloads that were 1 to 10,000 base pairs in size, and we quantified integration. There's no drug selection step here. We're just like seeing how many of the resulting colonies are positive. And you can see you essentially convert the whole population into now a genome containing the payload, and it works as well with the large payloads as the small ones, which again, we think will be important for potential future uses. I'm not going to say anything more about it, but we're teaming up with Harris Wang, who's in systems biology and studies the gut microbiome, primarily in animal models and mice. We're very excited, given all the data we have in bacteria, that we could now use these as a new way of doing precise genetic perturbations to bacteria. People have used random transposons for genetic screening, like TNC libraries. We think now the ability to land a payload at a user-defined site in the genome could open up some pretty exciting experiments. And in particular, we're doing experiments where we're doing that reaction in situ in the gut microbiome. So now you can think about targeting specific commensals or pathogenic strains, and either do things like eliminating a multi-drug resistance gene, or maybe adding new metabolic colonization factors as a way to either study the gut microbiome function or even target pathogen strains in this complex environment. But I'm going to talk for the last 5 minutes, hopefully, about some recent work we've published to leverage these systems in mammalian cells, where we think there's some real opportunities for solving some of the limitations of the existing CRISPR toolkit. So I showed this slide earlier, and I've got the world's expert in the room to school me and everyone else here on how darn complicated this repair step is after you make a double-strand break. This slightly more detailed cartoon is still a massive abstraction, but it makes the point that when you generate a DNA double-strand break in most eukaryotic cells, there are 2 or more primary pathways for repair. If you want to make a knockout in a typical gene editing experiment, you might be perfectly fine to get scars from non-homologous end joining. These are random insertions or deletions that can knock out the function of a protein coding gene. But often what you'd like to do is make a precise modification, either mutate a specific nucleotide or make target insertions. And here you need to now combine your nuclease with the donor template, and you need the cell to go through this alternative pathway of homology-directed repair. The challenge is that in general, the efficiency of this HDR pathway on the right, particularly in mammalian cells, is far less efficient than end joining on the left, which means you want to do some precise change. The majority product in your cells will be scars from indels. This pathway is also restricted to cells that are actively dividing, which means you can't easily do HDR insertions in post-mitotic cells. The efficiency of this gets even worse when your goal is to insert large payloads. We already talked about the fact that you get a heterogeneous mixture of products. And then you've got issues of triggering the cell's DNA damage response, which you may not want to do in certain clinical settings. And the fact that recent studies have shown that DNA double-strand breaks also cause large chromosomal translocations and deletions in a non-trivial percentage of cells. I'm not going to go to the details, but lots of attention on doing gene editing in T cells for cancer immunotherapy, where you might want to integrate chimeric antigen receptor genes while also editing or inactivating the TCR receptors or PDCD1. What has been seen in recent years is that you actually end up with sometimes up to 5 or 10% of cells where you're trying to make an indel knockout that actually become either aneuploid or experience large losses of genetic information at the site you targeted. And so again, from a safety profile, you can't easily remove those cells from a population before you might deliver these back into a patient. And so there's been a lot of push in the field to move away from nucleases because of the unavoidability of these kinds of aberrant products. And so there are a number of new editors that have been developed in recent years and used and developed by folks sitting in front of me as well, like Alberto over here. Base editors are tailored to making single base pair changes. Prime editors can be used to make more flexible edits. And then our goal is to develop these cast systems as an approach to make large insertions that could be kilobase or kilobases in size, particularly for gene insertion as either a research or a therapeutic approach. Just to go right to like a case study or test case for why we'd like to do this. Don't know why the slide is choppy. So this graph here is showing a couple hundred different recessive genetic diseases, and it's plotting a histogram of the number of known pathogenic gene variants that are found in patient cohorts for each of these diseases. So sickle cell is going to be way out here. Sickle cell is literally defined by one mutation that every patient with sickle cell disease on the planet has. And then you've got diseases like cystic fibrosis, which have actually hundreds to thousands of different mutations that all have a loss of function phenotype, which leads to similar symptoms, but the underlying genetic basis between patients can be very different, which spells trouble for clinical programs to correct mutations if you need to develop a tailor-made approach for every single patient mutation. And I might be going off slides for the last couple minutes because it's spinning. I got the spinning wheel on my computer. Microsoft is not responding. Restart the program. Sheila, do I need to like reshare here? I'm guessing.
No, you probably are okay. Well, you probably have to stop sharing. You're,
This is what we're having issues with earlier.
do the stop share first because you haven't restarted your PowerPoint yet.
Let me just see if this
There you go.
Oh, that's good.
And you also have a question.
and Zoom.
Yeah.
Okay. So with cystic fibrosis, my next slide is going to show, Our goal would be a universal strategy where you could, for example, cover all patient mutations by integrating the full length, well, how do I do this faster? Integrating the full length cDNA for the healthy copy of the gene that could, in theory, be universally applied. Wow, it's weird. This one slide seems to be the problem. There we go. Hopefully it's not going to. So here's like an approach that has been already validated in vitro. You can easily take a common patient mutation like F508 delta and use either HDR or base editing or prime editing to correct that mutation. Our case study of a type of goal would be, rather than the challenge of how you scale this approach for every one of the thousands of different mutations patients have, could we actually insert a full-length cDNA in the first intron that could essentially be applied to all patients with this disease, regardless of their mutation? So that's the type of application that we'd like to develop Cas systems to be able to do in the future. So do they work in mammalian cells? So the challenge, of course, as you hopefully already appreciated, is unlike Cas9, which is one protein and one guide RNA, we have this beautiful arrangement of components that lead to this really cool activity, but it's going to be a pain from an engineering perspective because we've got to get all those components working well in other cell types. So a lot of work went into getting this project off the ground, and it was brought to its first endpoint by two PhD students, George and Rebecca. Just going to make the point here, like one of the things we found early on is we got to get things trafficked to the nucleus, which means we have to add nuclear localization signals to all the components. We did this kind of haphazardly at first. We'll put it on every N terminus for all the components. It didn't work. Then we thought, well, maybe we broke the system by tagging it in this way. And so we went back to bacteria, and we now systematically tested different tagged constructs one at a time. And everywhere where you see like the numbers drop to 0 are cases where a single tag on one terminus of one of the components completely broke the system. So that meant we had to really go back and use these data to rethink how we're expressing and reconstituting things in cells. We used some experiments I'm going to skip over in the interest of time. We did some ChIP-seq to show that target binding was fortunately still highly specific in human cells. And the teaser here is we have not yet done the same kind of high throughput sequencing to look at specificity for DNA integration. That's actually ongoing right now. But in this first study that we published a month or two ago, we leveraged these different versions of Cas. So we had to go to a new version to get this first major bump in integration activity. We did some further improvements to hit 5% integration on plasmids in hex cells. Here's a control where we make a single point mutation and integration is dead. So we know all the genetics we learned in bacteria are still the case. And then again, I'm sorry that I'm going a little fast here, but we made one more improvement in the study to get to an engineered Cas version 3, where now we kind of rescued what had been a frustrating low efficiency at genomic target sites to reach levels of around 1%, and then showed that with this version 3, we could hit about a dozen genomic target sites in hex cells and hit efficiencies of DNA insertion of like a 2 or 3 kb payload of around 1%. So important first step, but clearly not at an endpoint where this is going to be useful. And so lots of ongoing projects in the lab and ideas for how we're going to further improve and increase the efficiency, which I'm just broadly categorizing here as how to express and deliver all the components, further mining, as well as some exciting work we're doing to evolve new variants, where we think that actually we can use experimental methods to isolate variants that are just hyperactive beyond the efficiency that nature gave us. And then lastly, using the structures that I showed we're doing some rational structure and function guided engineering to make changes that we think also have a good chance for further increasing the efficiency of the system. So I will just end there. Thank all the grad students and postdocs in the lab. And Ron, thanks again for the invitation today, and I'm happy to take any other questions here or on Zoom.
Want to handle your own questions?
I can do that. I don't know how to get to Zoom though.
Well, I'll ask you a question first. And you may want to repeat it for the people in the Zoom audience. So at the beginning, you mentioned that host factors are normally not involved, but they actually are involved, okay, often. And Steve probably knows about it more for human stuff. I know a little, I mean, if you think about lambda integration, the host factors are extremely important. I wondered whether an intermediate interesting step would be to ask, can I get the efficiencies that I see in bacteria in yeast, which has a simpler genome size compared to human cells? And it would tell you right away that you're missing something. And then you could start doing complementation in yeast from, oh, I need to add this host factor from bacteria to make it more efficient in yeast. And then you can monkey with your mammalian cells.
Yeah, so all amazing points, and I've got like an hour version of my answer to that. Actually, it's not an answer, it's a discussion, because I would love to show you some more data. But you're absolutely right, and we like kind of had to, what is the expression, to eat your shoe or something? Is that the idiom? Anyway, because we didn't write it doesn't need host factors, but I think we said host recombination factors.
Yeah.
We've actually now identified factors that are important for increasing efficiency that are in the bacterium, but not packaged by the transposon.
I mean, the good news is Vibrio vector
Works fine in E. coli,
great
yeah.
in E. coli. So it's some bacterial thing that's going to be.
Yeah, but long story short, there are some accessory factors that we're hoping to not need, but engineer around. And we did start in yeast. And we kind of just moved past yeast, but we actually did some experiments in yeast for a month or two, and we probably got some media from your lab. But that was years ago.
Is there a greater risk of off-target insertion out of the cell? Like, can it take itself out and mutate?
Excellent question. So for detailed transposon biochemistry reasons that were worked out on a related element many years ago, it's not thought that the transposase, it only excises when it finds a target site. Unlike, for example, piggyback, like you worry that piggyback, you have no way to control it going in and stopping. So you can get an insertion and then the piggyback transposase will just excise it right back out. These transposons only excise when they recognize and find the target site. So we think even in a human cell experiment where you had constitutive like expression over many days, unless there's another target site that the genomic insertion can like actually access in 3D space, it's not likely to excise after it goes in step one. But we are like, the truth is we haven't really done the right experiments to test that, whereas like we'd like to take a strain, a cell line where we've integrated it and then we got rid of all the factors, and then we take that and we put in boatloads of the transposase and track the persistence of the integrated transposon under conditions where we do long, week-long expression of the enzymes. So we haven't done that, but we don't think it's going to be at risk of getting popped back out. And then the other part of your question, just about increased off targets, it's definitely been shown with Cas9. You get more off targets with plasmid-based expression because you get much longer expression as compared to like a transient pulse of either mRNA or RNP delivery. So our long-term goals, if and when we're using it in mammalian cells for some downstream use, would be to deliver mRNA and the donor DNA, but mRNA for all the protein components so that we get kind of like turnover of all the expression material after about a day, so that we kind of only pulse it for as long as we need to get integration, but then the mRNA will degrade fairly quickly. But that's another thing that we're just at the early stages of starting to look at.
Say we have one question from the audience, and they ask, well, what type of cancers has T cell editing been applied to immunotherapy?
Yeah, good question. I'm not, maybe someone in the audience can save me. I mean, I think they're all like blood cancers for the most part, leukemias. Yeah. The same leukemias where like checkpoint inhibitors have been shown to be very successful. Yeah, so I think the companies that are developing CAR T cells are going after the lowest hanging fruit, which are, yeah, blood cancers where it's already been shown that like CAR T therapy without gene editing has shown efficacy. I think like solid tumors, solid cancers, that's still areas where there's going to need to be a lot more work.
And we're out of time. And thank you so much for doing this. This was great.
Yeah.
Absolutely great. And actually, thank you to the audience that came here.
Yes, thank you. Yeah.